Hello, everyone, and welcome to today's webinar, Fuel for Survival. We are thrilled to have you join us. Throughout the presentation, you can submit your text questions to our presenters by typing them into the questions panel at the bottom right of your screen. Please feel free to send your questions anytime. We will collect them and then address them during the Q&A session at the end of today's presentation. To maximize your webinar experience, you can participate using other toolbar panes and apps, including handouts, chat, and polls. Event handouts, which will include your certificate of attendance, can be found by clicking on the handouts in the apps. If you experience any technical issues today, please click on the help button on the lower left of your screen. And please note that today's webinar is being recorded, so after the event, you will receive a follow-up email with a link that you can view the recording. And without further ado, I would like to now introduce today's speakers. Tobias Kollmann, MD, PhD, currently is Professor of Microbiology and Immunology, as well as Pediatric Infectious Diseases at Dalhousie University in Halifax, Nova Scotia, Canada. Tobias is the CEO of the Born Strong Initiative, a global network of experts working to reduce adverse pregnancy outcomes. His expertise centers around immune autogeny, as well as maternal and early life vaccine responses, employing cutting-edge technology and analytics to extract the most information out of the small biological samples obtainable. And Nelly Amenyogbe, PhD, is a postdoctoral fellow in the Department of Microbiology and Immunology at Dalhousie University, Halifax, Nova Scotia, Canada. And her previous research showed how BCG, the vaccine against tuberculosis, reduces risk to die from newborn sepsis within days of being given. Her current projects build on these initial observations to identify how metabolism influences the immune response to infection, especially colostrum feeding for newborns, which may influence the efficacy of immune targeting therapies. These research questions are answered using preclinical models and multi-omics surveys of human, maternal, and neonatal immune responses. So now I'd like to turn it over to Nelly and Tobias to begin today's webinar.
Well, thank you for the introduction, thank you for the invitation, and thank you all for joining us online today. My name is Toby Kollmann, and
my name is Nelly Amenyogbe.
And we are absolutely pleased to be here to present to you on a topic that is very, very dear to our heart, namely fuel for survival. Today's learning objectives are to introduce the concept of the golden hour, to learn about WHO's recommendation to initiate breastfeeding within the first hour to reduce neonatal infectious deaths, to recognize the global challenges to that concept, to understand barriers that prevent half of the world's newborns from receiving colostrum, and to start delineating the unique properties of the mysteries around colostrum's life-saving properties. And in the context of all of these, to present to you innovative research that includes us, but many others, that are uncovering how colostrum actually works. The outline for today is going to be fittingly starting with me, focused on the problems. And
I will then swap speaker spots with Toby and talk to you about the solution. But for now, I will leave it to Toby to tell you what the problem is that we're dealing with.
This is a slide from a website called Our World in Data, that on the x-axis shows you age, human age in years, on the y-axis the percentage of death. And the different colored lines mark different decades that have been recorded. And while there has been clearly an overall improvement in or reduction in deaths throughout, everybody's born, everybody dies, so in the end this all adds up to 100%. But if you look at this distribution of deaths over the human lifespan, certainly it's going to creep up again towards the end of life, but the highest risk period to die is actually early in life. What do newborns die of? This is a graphical display, proportionally, roughly, from the WHO and the UNICEF, that shows the major categories, namely preterm birth complications being the major cause associated with death of newborns, intrapartum-related complications, sepsis, and several others. It is important to point out that within each of these categories, there's a multitude of upstream and downstream complex interactions. So this isn't just a single disease in each of these categories, but a multitude of causes. For example, in sepsis, Multiple studies, including this one done from Southeast Asia, have identified pathogens in a large fraction of newborn sepsis cases associated with death. And these pathogens are many, and they vary from region to region. Largely due to these complexities of the causes of newborn deaths, improvement, a reduction in neonatal deaths has not been sufficient to improve or to reach the goal of less than 10 deaths per 1,000 live births in the neonatal period by 2035. This graph again on the x-axis shows time in 5-year intervals. On the y-axis shows mortality rate per 1,000 live births. The green line depicts the under 5 mortality, so children under 5 years of age. And you can see in the large dashed line, and that's where we're currently tracking. The dotted line is where we'd like to be, but there's been a substantial improvement in the overall reduction of deaths for under 5-year-old children. If you compare to this, the line in purple, that's the global neonatal mortality rate. You can see that the improvement, relatively speaking, have been marginal. There's been an improvement, but nothing close. And we're certainly not anywhere on track to reach the goal of under 10, which is the stars in this graph, under 10 deaths per 1,000 live births by 2035. So to sum up the first part of the problem, that as neonates, neonates are the most likely to die. The causes for neonatal deaths are many. Interventions are very few. And with this, it is very difficult to improve the trajectory of neonatal deaths. And with this, Nelly, on to the solutions.
So our research program has been looking at these data for quite some time. And while we started off really focusing on understanding immune development or immune ontogeny of newborns, and through that figuring out what the most promising solutions might be, in a roundabout way, we came back to colostrum. And I'll give you the data that led us in that direction first. But first, let's visit the epidemiological data that supports our research focus. So immediate breastfeeding. Some of you might be familiar with some of the recommendations, but what's always interesting to us is to take a step back and have a careful look at the data that delivered those recommendations to us. The recommendation specifically that I'm talking about is initiating breastfeeding within an hour of being born. And some of the earliest findings that really painted a picture of why that's important are displayed here. In large observational studies nested within clinical trials that showed a clear relationship between the timing of initiation of breastfeeding and neonatal mortality, specifically due to infectious diseases. Now, these were predominantly done in low and middle income countries. A meta-analysis that I'll delve a little bit deeper into nailed this observation down. Those combined data from Ghana, India, and Tanzania. But the same trends are increasingly being observed in high-income countries, including the USA. And we have one continued reading for you here from The Lancet, published in 2022. So what do those data actually look like? Because here we were quite shocked ourselves when we looked at the observational data in quite detail. If we're looking here at the meta-analysis, first of all, the power of these studies is they were really able to look at a large number of records, so almost 100,000 newborns that were in their databases. And because in these study conditions, this was all nested into a trial evaluating the supplementation of vitamin A at birth by the NeoVita study group, they were able to look in quite good detail when newborns started to breastfeed, either within the first hour, from 2 to 23 hours of life, or from 24 to 96 hours. And if you look at the mortality rate between that compares newborns that initiated within an hour to later, from enrollment, essentially right at birth, to 28 days, the end of the neonatal period, the increased risk for neonatal mortality is clear. But even if you look at events that happen between 5 and 28 days, so now you're removing all those very early deaths that might be attributed to a large range of birth complications, the benefit is still there. But that's not it. If you take even a step further, now if you're looking from 1 to 3 months of age, you're still seeing the benefit. And again, even if you look from 3 to 6 months of age, the protective effects of early colostrum initiation are still evident in our data sets. So while the vast majority of births in the neonatal period do occur within the first week of life, the protective effects of early colostrum initiation seem to linger for far beyond the neonatal window. So if we look at this infographic here, which was done by the World Health Organization in conjunction with UNICEF, you can see how these data very clearly informed policy, even if you look right down to the interval, less than an hour, 2 to 23 hours, or 24 hours. What you're seeing is that the global recommendation now is that newborns should be breastfed immediately. And the data that inform these recommendations are the studies from the NeoVita study group, amongst others, that we just looked at together. Now, I think we all know that just because there's a policy in place, it doesn't necessarily mean that a problem has been solved. And of course, even if the best efforts were put in place for mothers to get the support that they needed, there would still be some newborns that cannot receive their mother's own colostrum for a wide variety of reasons. And some of those research opportunities Toby is going to talk about towards the end of our talk today. But we, as immunologists initially, and now we've had to broaden our scope of what could possibly be important other than the immune system alone, started to think about what could be the reason why breastfeeding early is so important. And one of the initial things that we landed on are the life-saving properties of colostrum. Now, for those of you that are not familiar with what distinguishes colostrum from mature milk, here it is in a simplified form by a nice little stress releasers that we got from Medela some time ago. But mature milk is a white substance produced in copious volumes that has a lot of energy to promote growth. Meanwhile, colostrum is a very yellowish substance produced in low volumes only for the first 2-ish days of lactation until a woman transitions over to mature milk. So it is definitely very distinct in its composition. This is why the first solution that we're focusing on is early initiation of breastfeeding, but specifically now conceptualizing colostrum as a fuel for survival in early life. And this is what we're investigating right now in our lab. So if colostrum could be a fuel for survival, what is it about colostrum? Now, we haven't figured out all the answers right now. This research is ongoing, but as a start, there's a lot already known about what's in colostrum. What are the major compositional changes? So the fact that mature milk is copious in volume and rich in fats gives it a yellowish color. The fact that colostrum is devoid of a lot of these fats, but instead is enriched in a lot of other immunomodulatory molecules is what gives it its yellowish colors. But of course, understanding the composition of a complex food like colostrum is different than understanding the biological mechanism through which these components act and for what aspects of neonatal development are critical for which component. And so the way that we actually started on this journey is by focusing on neonatal sepsis specifically. So in our lab, we oftentimes employ preclinical models to try to understand the physiology of neonatal responses to infection. And where we started was with a polymicrobial sepsis model that was initially developed by our colleague and friend, James Wynn, who is a neonatologist currently working at the University of Florida. And so he adapted a model that was primarily used in adult mice, where you take some of the intestinal contents of adult mice, make a homogenate out of a polymicrobial homogenate out of the substance, and use it to challenge neonatal mice. This model, which was originally published in 2007, is becoming increasingly popular to study both neonatal and adult sepsis in preclinical models. And we primarily utilize this model to understand, as was said in the introduction, how the BCG vaccine in early life was able to improve neonatal sepsis outcomes. But while we were doing this research, we started to ask ourselves the question, why is it that, given BCG aside, different pups, even with the same litter, can have such dramatically different outcomes to the same challenge dose? And so we took advantage to the natural variants that we observed in our experiments to look under the hood by collecting organs from these animals at a time when we can reliably predict whether they would survive or succumb. In our animal models, this would be for them to reach humane endpoint, and see what are the major pathways, immunologically and otherwise, that were different in their blood, their liver, and their spleens, for example. And what we landed on, these networks here were developed by our collaborator, Amy Lee, who's now at the Simon Fraser University. In the liver specifically, 3 biological pathways really stood out. The first being angiogenesis, or vascular integrity, so the ability of your blood vessels to grow, but also for the endothelial cells that make up your blood vessels to stay close together. And also metabolism of lipids, and specifically arachidonic acid and eicosanoids, which have many effects throughout the body, some pro-inflammatory, some anti-inflammatory, and some not yet fully understood. And so using these data, we actually started to select different compounds to see if they would improve sepsis outcomes. So the two graphs I'm showing you here follow pups challenged at about their 6th day of life with a polymicrobial sepsis, and we track their survival. So how long these animals, how long it takes for an animal to either reach human endpoint, or for the ones that survive who then recover fully from their disease. And here you're looking at % survival on the y-axis. And so first of all, arachidonic acid given orally shortly before challenge improves survival outcomes. So here you see the pre-treated pups that were given arachidonic acid compared to the ones that were not. Now, there are two compounds that are very important for your endothelial cells to close up their tight junctions. Tight junctions being the proteins that your cells use in order to maintain a tight barrier. Now, during an inflammatory response, we know that it is normal for tight junctions to be released. This allows immune cells from the bloodstream to exit the bloodstream and get to the tissues where they need to do their work. But for something like sepsis, where you have inflammation all throughout the body, if this process is not tightly regulated, then excessive vascular leakiness can become lethal very quickly. And two of the key proteins that your compounds that your endothelial cells need to regulate this process is a protein called angiopoietin-1, which signals for the endothelial cells to close up, and an amino acid called L-arginine, which is necessary for the enzyme that orchestrates this whole process to function. So first, looking at untreated animals here, in this particular experiment, the challenge was quite lethal. If you give either of these compounds individually, they rescue our experimental animals about 50% of the time. And if you give them together, they actually are very, very effective. Importantly, using the angiopoietin and L-arginine cocktail was even effective if these compounds were administered after the mice were challenged, meaning that they have therapeutic potential, which other things that we investigate, such as the BCG vaccine, would not operate in this way. It only works as a prophylactic agent. So now we had to take a step back, because I admit, at this point in time, even though we were so focused on neonatal immunity, we weren't really looking much at breast milk. And in retrospect, of course, this was a huge oversight in our part. But at this point, we had to stop and pause and ask ourselves, if these nutrients could improve outcomes, where could newborns be getting them from? Lucky for us, at the time, our lab was at the Kids Institute in Western Australia, also at the University of Western Australia, our affiliate institution, where Professor Valerie Verhasselt, who is a true pioneer in studying the relationship between breastfeeding and immune development, was also doing her research. And when we were exchanging our thoughts, the first thing she said to us is, well, what about colostrum? Because in her eyes, when you look at these immune compounds and how they change the immune response, the most important source of nutrition for a neonate is breast milk. And if we look at the compounds that I spoke on specifically, so arachidonic acid, L-arginine, or angiopoietin, they are found in breast milk, but they are found at much higher levels in colostrum than they are in mature milk. The other thing that we learned by working closely with Valerie is another way to study the effects of colostrum using preclinical models. So the model that she uses and that we now employ for our research is a simple model where as soon as pups are born, you cross-foster them either to another female mouse that has just delivered and is producing colostrum, or to a female mouse or a dam that delivered a week prior or 9 days ago and is producing copious amounts of mature milk. Importantly, while there are many species level differences between murine or mouse and human colostrum, the dominant trends in the macro micronutrients that shift are conserved. So you can see this yellowish color of the colostrum substance from early milk in mice, and then the characteristic yellow color of mature milk from the same species. So Valerie's team has been using this model for quite some time. And one of the interesting findings that they had is if you deprive pups of colostrum, they are nursed, they are well taken care of, and they are fed by the dams that are producing mature milk. But when you infect these pups with intestinal parasites or helminths, They have a higher burden of worms in their feces, also more eggs, and a key cytokine, interleukin-13, which is necessary to drive the protective response against helminth infections. This response is somewhat shunted in mice that don't receive colostrum. The other important piece of evidence that she was able to deliver by working with a colleague in Uganda is by accessing stools from children that are from 1 to 3 years of age, combined with the information of whether they were breastfed within the first hour of birth or not, they were able to show that that infants who are a year or older, who were deprived of colostrum in the first hour of life, the likelihood that they had a helminth infection in that time was significantly higher. And when we applied the model to our polymicrobial sepsis study, we found that the mice that did not get fed colostrum, even though they were growing at an equivalent rate, at least in our hands, to the mice that were fed colostrum, they had a dramatically increased susceptibility to polymicrobial sepsis, not just in overall survival probability, but also in the kinetics as well. These mice seem to reach their humane endpoint much faster than mice that succumbed that were also fed colostrum, and this is driving a lot of our lab's current research. What I will mention is this particular data is not yet published, but we are quite confident that this effect is reproducible in our hands. The other things that we were able to do is link the effects of breastfeeding to immune development in human cohorts. Now, this work is in its early stages, but the mainstay in our lab for quite some time has actually been understanding newborn immune ontogeny, both using preclinical models, but also working with human cohorts from around the world. And as members of the EPIC consortium, one of the key cohorts that we gained insight from was able to sample newborns over the first week of life. And from those data, we ran multiple assays from their blood samples in order to better understand how their immune system was changing. Now here, what I'm showing you is each point on this principal components analysis is one infant. And what you're seeing encapsulated here is a day of life. And what this figure is meant to illustrate is taking most measurements that you can derive from the blood. So all the transcripts, the cells, the proteins in the bloodstream, we're able to tell with a high level of accuracy how many days old a baby is, and in some cases, even how many hours. What that goes to show is newborn immunity is a rapidly moving target, which is perhaps why interventions early in life might have such a powerful effect. Because unlike an adult where you stay the same pretty much from day to day, for a newborn, every hour is a massive shift in immune development and ontogeny. Now, if we look at what are the other key sources of variance in immune development in early life, what we followed that up with is looking in detail at the cellular composition in the blood with a tool called flow cytometry. Now, of course, we expected that day of life was going to have most of the effect in overall composition of your peripheral blood cells, but breastfeeding came in at a close second. And second to that, for the babies that our team was able to determine when they initiated breastfeeding and how long the delay was, there was quite a strong negative correlation between the amount of hours for which breastfeeding was delayed and the degree to which their circulating pools of mature neutrophils declined. Now, this is really important to us because we know that neutrophils are a key cell type necessary for a robust response to sepsis. And having low neutrophil levels or neutropenia is a risk factor for poor sepsis outcomes. This is something that in the clinic is quite well recognized. So what we know about the solution so far is early initiation of breastfeeding seems to be very important. And we think that the feeding of colostrum specifically is going to be very important. However, from the epidemiological data alone, we had little knowledge of what those mechanisms can be. So now we're starting to take the next step and detangle those in our lab. And so far, what we know is if you take some compounds that are known to be enriched in colostrum, like arachidonic acid, angiopotin, and L-arginine, they are highly efficient at improving survival outcomes, both prophylactically and in some cases therapeutically as well. And so that leaves us with the next challenge, because again, we alluded to this at the beginning, even if we put our best foot forward to support every mother to initiate breastfeeding early, it is not going to be possible 100% of the time. So what are we going to offer for newborns that are not going to receive their mother's own colostrum in the window when they need it most? So this is all an ongoing investigation. We're very generously funded by an organization called Open Philanthropy, and which is how we conceptualized this Fuel for Survival project to begin with. We were also very fortunate to convince a global team of excellence to join our efforts, including some of the world leaders in understanding breast milk and its benefits for humans, such as Megan Azad, Lars Bode, who might be a familiar face to some of you, Valerie Rahasselt, and Fadil Hanan, but also to some of the leading edge informaticians that can help us detangle some of these signatures and complex data, like Nima Agiapur. And both Kevin Kane and Helen Barsosio, who agreed to collaborate with us to start to collect the samples and the information from a robust newborn cohort in Kenya so that we can actually mine the data and find the biological signals we think are important. Now I'm going to pass the stage back on to Toby. He's going to talk about a few more problems that we face, but also a few opportunities for us as a community to address this.
The first problem already mentioned by Nelly, if you take a look at the number of infants, of newborns that get fed within 1 hour of birth, it's pretty abysmal. This shows that of the newborns in Latin America and the Caribbean, only about 52% get fed within an hour. The numbers are similar, if not worse, in the rest of the world. In some areas down to 1/3, maybe 40%. If you combine this, the current problem is extended to the fact that less than half of newborns in the world get fed with colostrum within 1 hour of birth. The reasons for this are manifold. It's certainly not mother's fault. There's complex cultural and systemic hurdles that would have to be overcome to deal with this. But those are hurdles that can be overcome. Now, we can look at problems such as the number of infants who actually get fed on time, less than 1 hour, and look at this as a problem, or turn it around and make a problem into a tool that opens up opportunities. So here now, the problem for immediate breastfeeding. And for many of us who've been part of a birth process know how hectic and stressful the actual birth can be. Hard to imagine how breastfeeding within 1 hour has to be the top priority in that setting. So what is it about the timing of colostral feeds? Why is it so important? I'd shown you this graph before, where on the x-axis age, y-axis percent of death, to represent or to highlight the fact that the period of life that you're most likely to die per age unit is early in life. But it's not just in years as depicted here. This is actually true when you look at this in more fine granular detail as done here by a group in the UK who followed birth cohorts in England over several decades. And what they found is that it's not just in the first few years that you're most likely to die. It's actually in the first few days. Here again on the x-axis is age, this time in days, and on the y-axis again percent of death or rate of death per 1,000. It is the highest in the first 2 to 3 days, but not just the first 2 or 3 days. In fact, now the x-axis has been changed for the same data, and now looking at this per hour, you can see that the rate to die, the risk to die, is highest in the first 2 to 3 hours of life. Now this should ring a bell, because we just told you that the most important time for colostrum feeding is the first hour. Now put those two things together. These babies are likely to die, these newborns are likely to die in the first 2 hours of life. And at the same time, which argues that they're already sick when they come out, they're already infected at the time of birth. At the same time, if you give colostrum right then and there, the benefit is so pronounced, namely within the first hour of life. We have confirmed to our own studies that babies at birth can be diagnosed. Those babies that will develop sepsis later on can be diagnosed already long before they have any clinical signs and symptoms, so long before clinical presentation. So these babies are sick at birth already. And if you give colostrum then and there, it is likely that colostrum doesn't just have prophylactic benefits, the way Nelly had pointed this out, that last for months after colostrum deprivation versus provision, but it may also be therapeutic. This is, for those in the field of neonatal sepsis, this is a bombastic statement. Neonatal sepsis is highly lethal. There's very, very little that we can do once it has kicked in. And colostrum seems to be a magic bullet, possibly. This is a conjecture at this point, a hypothesis that we're eager to investigate further. But how can timing of colostrum, that's clearly important, be turned around into tools to assess other interventions and rank them in potential impact priorities? You've seen this graph before when Nelly introduced it, that the first hour is optimal. Is it really every minute, as this infographic shows from the WHO? If it is every minute, why is it every minute? Is it really the minute of life that matters? Or is it if you fed early, you're also more likely to be fed more colostrum or more frequently? So is it the volume or is it really only timing? And this is where potential for colostrum as an oral immune therapy to promote neonatal health has been recognized. This has been a practice where colostrum is collected from the mother post-birth through, for example, expression or pumping, and then administered to the baby that cannot otherwise feed, for example, a premature baby that's intubated. And it's administered to the baby either by a syringe in the mouth or via a swab, as it's often done, for example, in Eastern Africa. The benefit of this had been proposed in studies that showed reduction in necrotizing enterocolitis and potentially for late onset sepsis and others. A recent meta-analysis shows that there is no harm in providing oral immune therapy, so colostral priming, to those infants that cannot feed themselves. That the benefit, on the other hand, while there is trends towards benefit, the amount or the degree of impact of oral immune therapy does not come close to anything that Nelly had shown you previously from the cohort studies, the epidemiological cohort studies, where early feeding on mother's breast within the first hour of birth can reduce death by around 50%. So it's beneficial, but not to the degree. Oral immune therapy is beneficial, likely, but not to the degree that early feeding of mother's own milk at the mother's breast is. Why? That is something that could potentially be actually directly compared. Similarly, antenatal colostrum expression, and various other acronyms have been used, but ACE sounds pretty convincing to me, has been around for a long time, here reviewed by a group from Ottawa here in Canada, and this is a review more recently from the group in Western Australia. And overall, antenatal colostrum expression has been shown, again, to have very low risk. There is no signal, no significant signal of harm, including the potential risk for induction of early labor has been shown to not be so convincing as it was initially feared. The degree of benefit, although there is benefit for outcomes in terms of comfort of postnatal breast expression and the likes, the degree of biomedical impact of antenatal colostrum expression and supplementation with antenatal colostrum is also not reaching anything close to what Nelly had shown you. Again, about a 50% reduction of risk to die. So helpful, but maybe not as impactful as actual breastfeeding in the first hour of life has been shown. Now, this sums it up, that the potential opportunities that open up when you start looking at early feeding, namely less than an hour of birth, are tremendous. First of all, and most definitely, colostrum has prophylactic benefits, but it likely also is a potential therapy for established newborn sepsis. And beyond this tremendous potential to expand this into clinical application, low-hanging fruit through changes in policy are abound. The timing of colostrum feeding, as I mentioned, is crucial. Why is it crucial? Is it the volume? Can oral immune therapy be expanded? Can we do this more extensively? Can the volume be increased? Can antenatal colostrum expression be of any sorts and any approach? Can that help? And again, same thing, timing versus volume and the likes. Can colostrum be donated in milk banks? If so, how is it best stored? How can it be sterilized? How can this be expanded as a tool to supply benefit to many more? To summarize the key points that we have shown you today, and we're stressing the key points, is that immediate colostrum feeding at birth within 1 hour is the most effective intervention there is across the human lifespan. We know that colostrum feeding is not a medical intervention, but if you compare it to other medical interventions, any medical interventions across the human lifespan, nothing comes close to saving as many lives and livelihoods as early colostrum feeding. The practice of early colostrum feeding is definitely prophylactic, but also very likely therapeutic for newborn sepsis in particular. How does it expand to other infectious periods, let's say adult sepsis? There's no sign of any downside. No harm has been detected in any study that supported early colostrum feeding within 1 hour of birth. And there are many, many policy implications, what we call the low-hanging fruit, that without any further research, just implementing various approaches and algorithms to postnatal management, of how early colostrum feeding would provide substantial benefit. And lastly, while mother's own milk at the mother's own breast is definitely the best based on the current data, a nature-inspired colostrum substitute should be identifiable using the platforms that Nelly has just introduced to you, that should lead to something that can be offered to those who cannot immediately breastfeed. And with this, it really reaches equity around the world where many issues prohibit early colostrum feeding. And with this, both Nelly and I wanted to thank all of you again, first of all, for the invitation to present to you, for you to take the time to attend this webinar today. And we're thrilled and happy to answer any questions that you have now or in the future if you want to send us emails as a follow-up as well. Thank you.
Okay.
Wow, what a wonderful presentation. Thank you so much for joining us today and for that engaging presentation. We're really excited to begin the Q&A session now. So we're going to start the questions, but you can still continue to ask them through the questions pane in the lower right corner of your screen. So the first question that we have today is someone asking for the system review, looking at the timing of the first breastfeed. Did you take into account that those who have delayed breastfeeding may have been ill or require ICU admission, and that possibly being a reason for the delay, or possibly the reason for increased mortality?
Those are very good questions. Can you hear me? Excellent.
Yep, we can hear you.
Yes. So I would actually encourage you to pull the meta-analysis and read it in depth as well. I do know that as it goes with these kinds of analyses, they usually raise more questions than they answer. And so those were some of the key concerns involved. The nice thing about the initial paper that came out of just the Ghana site, they were kind of the first. The meta-analysis across all the sites followed sometime later, is they followed it up with some qualitative research where they looked at the barriers and facilitators of early breastfeeding initiation in their communities. And what they found wasn't necessarily neonatal illness. Some of it was just practice. Some of it was as simple as a mother would be tired, and so a nurse would take the baby elsewhere in a different room so the mother could sleep, and then the baby would be brought back sometime later in order for the baby to be nursed. So that level of qualitative data was not linked with the meta-analyses, so the same data set that was able to evaluate the mortality risks. So you can't stratify by that level, but given that this is a region where NICU care is hard to come by, it's kind of low resource settings, a lot of births that go home early on. It wasn't entirely a preterm phenomenon either. So this isn't only very small, vulnerable babies that are the ones that are dying out of delayed colostrum. And so through adjusting their analyses for as many confounders as they could in such large data set, they weren't able to find any other compelling reason why the early initiation itself was not protective, but perhaps just a signal of some other illness or extreme prematurity or maternal illness and the likes. That's a very good point because these are observational data. And so all of that likely does play a role for some infants. But again, those data are not able to disprove that the early initiation of colostrum on its own seemed to still be important.
So Jess, you're muted.
Oh, wait, thank you. There's so many great questions coming in. Thank you. But I also want to address, there's been a couple questions in the chat about the handouts for today. So if you click on the app section at the bottom right of your screen, there is a handouts link in there, and you can download a PDF of today's presentation. So please be sure to do that. All right, our next question. So someone says the data that you shared during the and early on in the presentation, it says it seems like it's a big time range from 2 to 23 hours. Was this accurate in the data that you had shared, or did she misinterpret it?
It is a wide time range. So these are the data as they reported, and you can see that those time intervals have been kind of adopted by the infographics by the World Health Organization, UNICEF, as well. Though, I mean, given that we approach all this stuff as experimentalists, that's one of our first questions as well. Could it be the first 2 hours? Could it be the first 3? And when we were at a conference in San Diego at the Human Milk Institute some time ago, and we had a chance to exchange thoughts from a very multidisciplinary audience, including many people that are kind of in the front lines of giving women the support they need. The feedback is, it would be very super if 3 hours was fine, because there's a lot of things that happen during the birth process that could make the first hour very hard to achieve in some situations. So was it accurate that it was binned in that way? I could say with confidence, yes. And again, these were not studies that we had any direct involvement with ourselves. This is just kind of the epidemiological data that we're looking to help interpret our biology. But one of the things that we're starting to ask experimentally as well in the colostrum deprivation model that we have is how critical that first hour is. The other great thing is now that we've picked up on this through our involvement with some of the human cohorts that we work with, including the AGREE trial in Kenya, we are now capturing that information at a much more granular level. So hopefully we could start to tease that apart a bit more than it has been published to date. I would actually say that this is a call to action for any of you guys that have the position to do retrospective chart reviews or prospective data collection on your wards. I know there's plenty of data that's sitting in sheets that's not been actually analyzed and put out there. If we kind of prioritize that as a community, I think some of that insight could be gained from data that already exists, has just not been analyzed in that way yet.
Such a great call to action. We love it when our webinars help with that engagement afterwards. Our next question, someone asks if there was any dose dependency looked at for colostrum with the protective benefits.
That's a very good question. I think Toby had alluded to that possibility earlier, actually. If babies get colostrum sooner, they also get more colostrum over the course of the day. Is that helpful? The short answer is we don't know. That is a research gap.
Thank you. So the next question that we have, someone asks, can the mouse model be used to test whether the amount of colostrum is important? So kind of building off of this last question, and wondering if maybe pups could foster with dams at different days and not just early and mature. Similarly, challenge pups could feed on new dams to maximize their exposure when symptomatic. So any insights there?
Very good questions. I mean, I'm happy to take them all. Yeah. So is it just the 9 days that's the magical window? Again, I think a lot of this, we're kind of in the midst of this right now in our lab. So in a year from now, we should have a wonderful paper that hopefully answers all of these. But for the Hellman story that Valerie had published some time ago, which I shared with you as well, she did evaluate whether the same kind of defects in a part of the gut immunity that allowed these helminths to persist was present if they cross-fostered to dams that were just finished producing colostrum, so like day 3-ish. And the phenotype was still there, meaning that even if you cross-foster to moms that have just transitioned out of colostrum, the effect on the immune system that allows makes it harder to clear helminth infections is still there. Now, in terms of sepsis, we don't know, but that's one of the questions we have. Another one is it's not just cross-fostering to a different mom, but if you delay feeding of any feeds for several hours, how is that going to affect our phenotype? All lined up, and things that we're hopefully going to be able to deliver insight on. The question on, is it possible to control more stringently the amount of colostrum a pup gets? Now, that's a very good one. And there's a few angles you can approach this. One, we were actually conceptualizing in our lab, could you make a synthetic feeding device for pups that closely mirrors a dam? Right now, it's all theoretical, but maybe if there's someone with the technological expertise that would like to work with us on that, we can develop this as a preclinical tool that could be deployed to any lab that's looking at this. The other solution, of course, is to not use mice. So we do work with a great team of investigators led by Nelly at the University of Copenhagen, and they run large animal models to study newborn metabolism as well. And so if you actually do a literature search on the benefits of colostrum, amongst the first to recognize and know of that benefit is actually farmers. Interestingly, I was doing a public outreach event here in our city last year, and it was to talk about colostrum and its benefits. And as people would come over, I would always ask, who knows what colostrum is? And one of the few people that did was a man who grew up on a farm, because farmers know if you give animals colostrum, they seem to do much better. And so, of course, their research environment is much more stringent than that, but there are ways to give controlled amounts of colostrum in other experimental setups that we might not always necessarily be able to do in our preclinical models. I mean, for those that don't do mouse work, a neonatal mouse is about that size. So it limits some of the things that we're able to accomplish in that system while opening up many avenues for others.
Great, thank you so much. Our next question, someone asks if there's a difference in outcomes if the baby latches directly for colostrum versus if the colostrum is collected manually and fed to the baby by feeding tube or syringe or in another way.
Do you want to take that one?
Sure. The short answer is we don't know, because that's not been directly compared. What we do know is that oral immune therapy, so the amount of, let's say, up to a maximum of 2 milliliters, which is the highest volume I've heard being administered to newborns, very slowly, obviously, by a syringe, versus a sponge wiped, soaked in colostrum and wiped in the baby's mouth. So going from 50 microliters to maybe 2,000 microliters. That range on its own has not been investigated. If there's a difference within that low range. We do know that, and that's what we had pointed out before, that the clinical benefit of oral colostral immune therapy versus feeding on mom's breast is several logs different. So the clinical benefit is much higher from the data that is out there if the baby feeds on mom's breast than if there's oral colostral therapy. But that does relate to several questions that I've seen pop up in the chat just now in terms of confounding cause with correlation. So in the human studies, we cannot have anything, have a study set up that allows us to control for this baby is sick and therefore goes to the NICU and therefore doesn't feed as much and as well on mom's own breast versus this baby who will remain with the mother and feed on mom's breast. So these cannot be controlled in a matched manner. Obviously, this is always going to be confounded on the human end for obvious reasons. But in the mouse model that Nelly introduced to you, the preclinical model, as well as the pig model that you mentioned with our collaborators in Copenhagen, this is controlled for, where we do check specifically how much, what volume, when, and who, and the challenges, or in this case, infectious challenges, are also controlled for. So we know from the preclinical model where we are able to dissect cause-effect mechanistic relationships that there's a direct correlation between the colostrum timing of colostrum feeding and outcome, independent of how sick the baby was at the time of randomization, the pup in this case. What we've not been able to do, which is what Nelly just addressed, is to address the issue of volume differences in that context. So timing we've begun to address this, and we soon hopefully will address volume as a variable in there. And with the preclinical animal models, we will be able to assess cause-effect relationships and come up with direct mechanistic connections, which is not going to be able under any circumstance with the humans, where it will remain observational and with that always subject to confounders.
Great, thank you so much. Someone else has asked us, you had mentioned about colostrum collected antenatally being less effective. If you had to speculate or had an idea of why this would be, what can you tell us?
So again, the short answer is we don't know. And that always is going to be the first answer right now, because the amount of, or the lack of knowledge about colostrum, despite it, and I'm going to repeat this one more time, being the most effective intervention that we know of across the human lifespan, the lack of knowledge is astounding. The fact that we only now are starting to look at this is really just, it's surprising. But to get back to this, what there is, Valerie had Valerie's theory, and I think that is a good one, is that each mom produces the exact composition, constellation, whatever, of the breast milk that the baby receives. So mother's own is always going to be very peculiar and specific for the baby, for her baby. And so I think antenatal colostrum expression, even if it's the same mother and the same baby in the end, it is not at the same time that the baby actually is receiving the colostrum that was harvested, is not produced at the same time that the baby is receiving it. So it could be, and again, this is complete conjecture at this point, it could be that what you need is what the mother produces at the time of that moment. That is by far the most effective. That's, again, conjecture at this point. We don't know that. It makes some biological sense, if you wish, from teleological or evolutionary perspective, but there is no proof for any of this. This is absolute conjecture.
Yeah, and I think what I would add to that is, if we look carefully at the evidence for ACE, a lot of the times the problem lies in heterogeneity of different studies. So I think why the first hour effect came out so clear is because, again, the studies that produced those data were not designed to evaluate early breastfeeding initiation. They were designed for a completely different purpose, but what it created was an environment where data was captured in a very systematic way from a very large number of neonates. But one thing that often is hard when we look at many small intervention studies is there's a lot of nuance, but maybe important differences in their design. Like, when was the antenatal colostrum expressed? Where exactly was it stored in the meantime? And when exactly was it given to the neonate? And then what's the sample size in those pooled studies? So I would think that a part of the problem is we've started to study ACE and its relationship to establishing breastfeeding more successfully. But we now also need to start studying ACE as a colostrum supplement for its effects on early neonatal outcomes. And then we have to think about what those outcomes need to be, because mortality as an outcome could work, or at least would have worked, in low-income settings at the time that those papers are published. But in high-resource settings, we need to think of other clinically relevant endpoints to employ in the kinds of care settings that we have. So I think a part of the we don't know is because we have not yet investigated it in a rigorous and systematic enough way in a sufficient amount of events in order to have a clear picture of what works. And so one of the questions we actually had asked is ACE equivalent to colostrum that you produce postnatally? And that's one simple question that could be answered by the community. We're starting to talk to other researchers that might be interested in collaborating with us on that. We know labor will induce a lot of changes in the bloodstream of a newborn. There's a lot of proteins that are made and transferred through the placenta around active labor that seem to have really important roles for the neonate. And you don't see those in the same way for babies that are born through cesarean section, especially when labor was never initiated. And so that leaves room for some of those same changes to affect colostrum composition as well. But again, right now, it's just a big research gap.
That's great. Well, thank you so much. I can't believe that we're almost to the end of our hour. I think we'll have time for one last question here. So someone asks, can we assume that donor colostrum milk would be an adequate substitute for mother's own colostrum if it's not readily available within the first hour after birth? Granted that the donor colostrum is available to mom and baby in that time period.
I'm going to start this answer with the same beginning answer that I gave before. We don't know.
We can't guarantee it.
Yeah, no. So I think that this kind of connects a little bit to the answer before as well. It is possible, and again, we don't know that, it is possible that mother's own is the only intervention that actually will have this incredible benefit that Nelly was pointing out. It's very possible. So it could be that any form, either antenatally expressed or donor milk or whatever you want, that none of those may have the same impact. We don't know that. And I think these are all, as Nelly said, given what a tremendous benefit this is. And we're only looking at hard outcomes like life versus death in these studies from Ghana and Tanzania and India and the US. But if you look at this across a multitude of different outcomes, from NEC to late onset sepsis to developmental growth, and there's so much that we know is associated with this early feeding. And we don't know most of the questions that you are all asking us. We don't know, not just we, but we as a field, we don't know. So I think what this really points out, despite the prominence of the impact of early colostrum feeding, the lack of knowledge about this is, I would just say it very flat, is unacceptable. I think we as a field own it to ourselves and the rest of the world that we work together to find out.
Yeah, I think so. What I would add to that is because we think it might be possible to, through mechanistically understanding how colostrum works, to figure out what can be given in its place. And I think when I hear a question like that, it's coming from a place, so what can I do now, right? And there's, say, a child is born, the mother is unable for a multitude of reasons to supply that colostrum right then and there. So what can I do now? Now, just because we can't say with certainty that donor colostrum is going to work, because there's no clear randomized trial that's going to tell you. My question is, do we know that there's a risk? If we cannot safely conclude that there is, but there is a chance of a benefit, I would make the decision and give the colostrum that's available. Now, I think the first thing for the healthcare professionals to establish is there any potential for risk? And of course, when it comes to donor human milk, a lot of research has been done to make sure that it's being given in a safe way. And so in the absence of risk, if there's a chance of benefit, given what's at stake here and given how how strong the signal of EBF has been, I think we're at the point where we should go ahead. We should also go ahead and generate the evidence that we need, so we are equipped with the knowledge to know what we need to do. But we also have to recognize that that knowledge is going to materialize overnight. It's going to take several years of careful work. And I think to put together some recommendations to the best of our ability from what we know about colostrum, so that people are equipped with some knowledge that they can use today, is something we should also be able to do.
Wonderful. Well, thank you so much for these insights and answering all of these questions. This has been wonderful. So as we close today, we certainly want to remind everyone and just continue again to tell you thank you for sharing this knowledge with us. As we leave the webinar and close, you're going to automatically be redirected to a survey about today's webinar, and we really appreciate you just taking a few minutes to complete it, as we really value your feedback. And you will also receive an email after the webinar that will have a link to the recording, and then you'll be able to rewatch it if you would desire. So on behalf of Medila's Medical Affairs team and our presenters, thank you so much for joining us today, and we hope you have a great rest of your day. Thank you.
Thank you all.